Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for your different examination. So this is the most important video and this was the most requested one by all of you, right? To provide the basic information about the advanced instrumentation, right? Like you want to know about how a specific technique is used and what is the application of that particular technique. So I thought to made a video right from scratch. So if you are new at my channel, do subscribe it to get different videos related to life sciences, be it for competitive examination, be it some job related or training opportunity kind of video, guidance, research guidance, or anything. So do hit the subscribe button, and if you like the video, do hit the like button as well. Now this will be a series related to PCR, as without PCR invention, the biomedical research field would have probably collapsed, right? Because if we talk about the confirmed diagnostic part, so without PCR, without molecular diagnostic part, we cannot do so. So this will be the first part where I will be providing you the basic information about the PCR, and then I will be providing the part 2, in which I will further discuss the components which are involved in the PCR reaction. And after that, we will be dealing with different variants of PCR. So this will be a wholesome video and most important one if you are targeting any examination conducted by ICMR, CSIR, DBT, or any examination, right? So this PCR basics video, this will be the part 1. So what is PCR? As everybody knows that PCR stands for polymerase chain reaction. So mind it, this is a chain reaction. It means there may have certain steps like first step, second step, and third step, and then these steps are repeatedly like repeating. These are repeatedly repeating and to have certain outcomes. So this is a chain reaction. Like after the first step, you will be going towards the second. After the completion of the second, you are going towards the third. And likewise, this cycle is repeating constantly. So this PCR is like that chain reaction. It is polymerase chain reaction because there is one enzyme, that is DNA polymerase, which is responsible for like multiplying your one DNA copy to even millions and billions of it. It is as equivalent as you are photocopying a single page in like 10 copies, right? If you are having like 10 student in a class and you are having only one, your teacher will do what? He or she will do photocopies of that and he or she will distribute to every student of the class. So likewise, PCR is nothing but a DNA photocopy machine. Now, who is the face behind this like excellent invention, excellent discovery? So it is Carey Mullis in 1983 who developed this technique PCR and also awarded Nobel Prize in Chemistry in 1993, right? So this technique is specifically responsible for amplifying a specific DNA sequence exponentially in vitro, right? We will see. So this PCR is frequently utilized laboratory nucleic acid amplification technique nowadays. Without having this technique, the medical sciences will like collapse, right? So just after the discovery of this one, it has like very basic PCR we are having conventional one, and after that nowadays we are having like latest PCR from manual conventional PCR total date we are having automated PCR. So we are having so many versions of PCR available right now who can offer so many applications in the biomedical field. So basically, this PCR utilizes a Taq polymerase. Why not normal polymerase, which we can use in this? Because we operate this particular reaction at such high temperature like 95-degree centigrade. So at 95, normal polymerase as enzymes are very much sensitive towards the high temperature. So only Taq polymerase, as Taq is Thermus aquaticus, the organism from where the enzyme is extracted. So therefore, it is capable of withstanding that high temperature that is 95 degrees Celsius. So for that reason, we have used the heat stable thermostable polymerase for the operations of PCR, and also without that PCR would not have been possible, right? So this technique targets the specific DNA fragment within a sample. Suppose you are having certain DNA sequence, right? Let me use different color pen. So suppose you want to amplify this particular region. It is your region of interest. ठीक है? Region of interest or gene of interest. So you want to amplify this particular segment. So what you will do, what technique you will use. So you will design primer. So primers are what? Primers are short oligonucleotides, basically 20 to 25 nucleotides long. So it will be complementary to your gene of interest. Suppose your sequence is ATGCTTA, then your primer, this is your source का sequence. So gene of interest में आपके ये sequence है. So you will be designing a primer whose sequence will be like this way. A will bind with T, T will bind with A, G will bind with C, C will bind with G, then A, then T. So if this is your primer and the above one is your sequence or gene of interest, so this primer will go and bind to the gene of interest and then it will be amplified and then you will get the gene of interest with you. So it is based on the complementarity principle, right? We all know that A will bind to T with 2 hydrogen bond and G will bind with C with 3 hydrogen bond. So as I told, Taq polymerase is favored for its thermostability. So after the binding of the primer to your gene of interest to the specific site, it will be elongated further through the help of the primers, right? So it is acting as a gold standard for different diagnostic tool, be it a bacterial, fungal, viral infection, or any other thing. So this is the most important one. These three cycles are repeated over and over so that we can get this amplification. So now next part, I will now let you know the most theoretical part of this like PCR. So PCR always begins with the extraction of small nucleic acid sample that is acting as your target, right? Target or sample sequence. ठीक है जहां पे प्राइमर बाइंड करेगा so it can be a DNA, it can be RNA into a reaction tube. That reaction tube is placed in an instrument known as thermal cycler. That instrument is known as a thermal cycler. See, this is a variant of a thermocycler. Most of the thermocycler looks like this. This is provided by different brands. You can see this is from high media, this is by applied biosystem. So the instrument used to kept those reaction mixture tube is known as thermal cycler. It is a device that regulates the temperature and duration of each step in the cycle. You can feed as per your own requirement, right? So see, it basically involves three steps. So as I told, you need a small nucleic acid sample. You can see here, as I told, this is your gene of interest. So gene of interest to आपको चाहिए होगा, that is acting as a target or the source DNA sample sequence. And after that, you will be doing the three steps. The first one is denaturation followed by annealing and then primer extension, right? So during denaturation, DNA is heated to 95 degrees Celsius to disrupt the hydrogen bond. So as I told, you have to amplify this particular region. So what you will do, you will be firstly, you have to convert the double stranded DNA into the single stranded. How you will do it? By destroying the hydrogen bond. And how the hydrogen bonds will be destroyed? By heating. ठीक है 95 डिग्री पे हम हीट करते हैं तो ये जो डीएनए है वो सिंगल स्ट्रैंड हो जाता है. अब यहाँ पे आपका प्राइमर बाइंड कर जाएगा. ठीक है सो यही होता है लॉजिक. यहाँ पे आपका प्राइमर आके बाइंड करेगा एंड देन it will be further extended in the extension step. So this is the like most important step of the PCR. So after immediately after the denaturation step, annealing is there. Annealing follows immediately, cooling the denatured strand to a temperature typically ranging from 55 to 72 degrees Celsius. जिस पे primer bind होते हैं आपके. Primer will be like forward primer and reverse primer. And after that, your extension step will be there. Now, what should be the annealing temperature? So annealing temperature is the most important one. It varies in between 55 to 72 and it depends on the primers, right? So as I told, what is primer? Primer is about 20 to 25 nucleotides long and primers offer like during the annealing step, primer bind to the complementary sequences on the single stranded DNA by pairing their 3' end. ठीक है, to the template strand providing a starting point for DNA synthesis. We know that 3' OH हमें चाहिए होता है synthesize करने के लिए आगे का strand. So primer का role यही होता है and the annealing temperature depends on your primer. So जो final step होता है that is at around 75 to 80 डिग्री सेल्सियस, ambient होता है 72. ठीक है, 72 डिग्री में आप फाइनल एक्सटेंशन करते हो. Then you put your reaction tube. I will also let you know the exact mixture of the like one reaction in the next part, in the next upcoming part. As this is the basic one, I will provide you the information regarding what exactly is PCR, why we do it, how we do it. Everything will be provided to you. Like basic से हम advanced की तरफ जाएंगे because everybody don't know about PCR. So see, repeated cycling results in amplification of the multiple copies of the target DNA in about 30 to 40 cycles, and after that जैसे ही 30 to 40 cycles होता है, आपके DNTPs एग्जॉस्ट हो गए हैं, आपका जो प्राइमर है एंड बाकी जो चीजें रिएक्शन मिक्सचर में आपने डाला है वो डिप्लीट होने लगते हैं, एंड ऑल्सो एक्यूमुलेशन ऑफ बाई प्रोडक्ट, रिड्यूस्ड एंजाइम एक्टिविटी एंड प्रोडक्ट टेम्पलेट कॉम्पिटिशन. सो इस वजह से क्या होता है, इम्पैक्ट आता है, इफिशियंसी कम हो जाती है आफ्टर 30 to 40 cycle. Now we are having some inhibitors that inhibit the PCR, right? So what it is proteinase K, phenol and EDTA, which are the inhibitors of PCR, right? So after the PCR amplification, we run a gel that is known as agarose gel electrophoresis, and we stain it with ethidium bromide to visualize the bands. And the gel is observed under ultraviolet light. Trans eliminator पे देखा जाता था पहले. Now we are having like gel documentation system, which is very easy, and it is very important to observe the for the visualization of DNA, to observe the DNA. So what we do in gel electrophoresis. So gel electrophoresis is basically for the separation of DNA and RNA based on charge to mass ratio. Suppose this is your like gel, this is the like negative cathode and positive one, and you have these wells here. Then the higher molecular weight will stuck like here over here, and the light in molecular weight will like go run more. यानी जो heavier रहता है वो slow run करता है, and जो lighter होगा वो migrate करता जाएगा. So this is how it is separated on the basis of charge to mass ratio, right? So as we can separate nucleic acid through agarose gel with different percentage of agarose depending on the molecular weight of the products, and we are having protein also. Protein can also be separated on the polyacrylamide gel that is made up of acrylamide and bisacrylamide cross-linking. So this is a vertical electrophoresis in which you separate protein. So apart from this gel electrophoresis, we are also having some blotting techniques that I will let you know later. Blotting, the basic one, southern blot is for DNA, northern blot is for RNA, and western is for protein, right? So what is the troubleshooting part here? If you are getting smeared DNA, then it is that you have degraded your sample. If you are getting weak bands, you can have low loading concentration. इसका मतलब ये है कि DNA का concentration low है, you need to load more. या फिर प्रॉपरली आपके लोडिंग डाइस काम नहीं कर रहे हैं, और नॉन स्पेसिफिक बैंडिंग मिल रहा है, मतलब आपका जो प्राइमर है वो नॉन स्पेसिफिक है, कहीं भी जाके छोटा सा भी उसको ओलिगो मिल रहा है तो वो वहाँ बाइंड कर जा रहा है, बिकॉज वी आर हैविंग रिपीटेटिव सीक्वेंस ओवर आवर जीनोम. सो इसीलिए प्राइमर जो है वो काफ़ी स्पेसिफिक होना चाहिए एंड इतना लॉन्ग होना चाहिए कि वो जाके सफिशिएंटली आपके टारगेट पे ही बाइंड करे. अननेसेसरी जगह पे जाके बाइंड करने की वजह से एम्पलीफिकेशन जो होगा वो फॉल्स एम्पलीफिकेशन आपको आउटकम्स दिखेंगे. So this is the way how we prepare the agarose gel, right? We prepare the buffer, right? 1xTA, and after that we take agarose as per the desired concentration, 1%, 2% or whatever, and then we heat it to properly mix this agarose, to properly solubilize this agarose in the TI buffer, and then after certain cooling we pour it into the gel casting tray with having a comb for the wells, and then we load our sample. So this looks like this one. This is your well made from different combs. See, this is the sample wells. You load your samples here. This is the negative electrode, black one, and this red one is the positive electrode. And Y negative DNA is negatively charged, so it will come towards the positive, right? So you will load your buffer solution. It has some ions which will provide electricity. So direction of movement will be like this way, right? You have one power supply where you provide the desired volt at which your gel will run. And this is the electrophoretic tank and the gel casting tray which you will put into that. And this is the way how you load your samples. Now the samples are mixed with the loading dye. What is loading dye? I will let you know. See, if you want to prepare a mini gel, let's suppose 50 ml at 2% concentration. Sorry. If you want to prepare like 50 ml gel at 2% concentration, then you will weigh 1% agarose. You will prepare 50 ml 1xTA and then you will dissolve it, microwave it so that your agarose will fully dissolve, clear, no such particles remains. You will swirl and check and then you will cool it down and you will add etherium bromide, right? For staining your DNA. To visualize your DNA, etherium bromide, as it is carcinogenic, so a lot of care is required while handling etherium bromide, right? So for this reason, we do not discard our gel anywhere, like in the sink or anywhere, because it has a carcinogen that is etherium bromide. So the concentration is 0.5 microgram per ml. So you will use it, and after that, you will place your comb in the casting tray, and you will like pour all of your gel which has etherium bromide properly mixed in it in the casting tray, and after waiting for about half an hour your gel will be solidified and you will pour 1x running buffer and you will load and run the gel. For loading, 6x loading die is used, which consists of some tracking die that will help you to visualize that at what extent your gel has run, क्योंकि हमें पता है ट्रांसपेरेंट होगा तो हम देख नहीं पाएंगे. यू कैन सी हियर, ये ब्लू कलर नहीं होगा तो कहाँ तक रन हुआ है वो आपको दिखेगा नहीं. सो फॉर दिस रीजन वी यूज नाउ what volume you use. If you are loading 5 microliter of your DNA sample, then you will use 1 microliter of your loading dye, so total will be like 6 microliter. ठीक है? So this is how you can visualize through your gel documentation system. This is a ladder both side and you are having certain samples who are amplified. So these are the things. Now, what is the role of bromophenol blue, xylene, cyanol, glycerol? These are the components of loading dye, right? So bromophenol 0.25% we use, which act as a tracking dye. It migrates at the same rate as the DNA fragment in 1% agarose, help to monitor gel progress. Okay? Without a color, we will not be able to locate that at what extent the gel has been run. So xylene cyanol is also a tracking dye which migrates with 4 to 5 kb DNA fragment, useful for checking migration of. For larger fragment, we use xylene cyanol. Like see, bromophenol blue will migrate fast. Xylene cyanol will move slow because it is for larger fragment, and we know larger fragments migrate slowly because of the molecular weight. So you can see it is around 300 base pair. It is around 4 to 5 kb. So run near the bottom of the gel, stays near the mid gel. यानी ये आखिरी तक रन होके पहुंच जाता है क्योंकि lighter in weight है. So use when analyzing small DNA fragments around 100 to 1000 base pair. Use when analyzing larger DNA fragment 1 to 10 kb like plasmid digest or genomic DNA. So for that we use xylene cyanol. ठीक है. What is glycerol? Glycerol make the sample dense. जैसे आप जेल में लोड कर रहे हो, where it is, जैसे अगर ग्लिसरॉल नहीं होगा, लाइट इन वेट होगा, तो ये वेल से स्पिल हो जाएगा. टू प्रिवेंट द स्पिलेज ऑफ योर सैंपल, डेंसिटी चाहिए, डेंसिटी इज प्रोवाइडेड बाय द ग्लिसरॉल, राइट? We are having tris HCl as it has the buffering capacity to maintain the pH during the loading and electrophoresis. EDTA is for it chelates the magnesium ion, which is the cofactor for DNases, therefore preventing DNA for enzymatic degradation. EDTA अगर आप add नहीं करते हो, so what it will do, it will like uh, act as a cofactor for DNases and DNases will degrade your DNA, right? And double distilled water we use to make up the volume, right? So this is the composition of your loading dye, which you use only 1 microliter for 5 microliter of your sample. So this is how you can see xylene cyanol is here and bromophenol is here, right? In 1% agarose gel, right? So this is how you can see. There are so many different size of electrophoretic tank and casting tray available, like so many different capacities are available, smaller, the medium one and the larger one. This one is the larger one. You can see so many samples are loaded here. This is the electrode which are placed in the power pack, right? So after that, uh, these are the basic thing which I have provided to you. Basic principle works on the complementarity and the semi conservative replication. यानी एक आपका सोर्स डीएनए रहेगा, एक न्यूली सिंथेसाइज्ड रहेगा, एंड टेस्ट ट्यूब में ये होता है इन विट्रो, अनलाइक द रेप्लिकेशन जो हमारे सेल्स के अंदर होता है इन वीवो। एक्सपोनेंशियल एम्प्लीफिकेशन कंसेप्ट क्या है? डीएनए डबल्स फॉर्मूला होता है n इज इक्वल्स टू n नॉट इंटू टू टू द पावर n. सो n नॉट इज द इनिशियल डीएनए कॉपीज एंड small n is the number of cycles, how many cycles you are running. So after 30 cycles you can have 1 billion copies, right? So now see why PCR is needed: to detect very small DNA quantities for disease diagnosis, for gene cloning, forensic science and research. Real life example, COVID was detected through RT-PCR. So it is very, very important. And key features of PCR include high specificity, right? Target DNA only को ही amplify कर सकता है, like पूरे stretch, इसमें से आपको सिर्फ इतना stretch चाहिए तो आप उसको भी amplify कर सकते हैं. So it is very sensitive and specific. It is quick, few hours में आपको मिल जाएगा, and it is fully automated. You don't need to do anything. Like आप जो reaction mixture prepare कर रहे हो, now we are having fully automated system. So those are of course expensive, लेकिन जो preparation reaction tube का है उसके बाद आपको manually कुछ नहीं करना है. Everything will be done by the instrument. So these are the applications of the PCR. So it can detect mutations and modification of the DNA, right, by designing primers. Like if you are using wild type primer and if it is not amplified, it means there are certain mutations in your gene of interest. You can identify different disease you can identify, be it a viral disease, be it a bacterial fungi based disease, bacterial disease. So it can also detect pathogen in the sample, right, by specificity. क्लोनिंग के जितने भी एक्सपेरिमेंट्स हैं आप कर सकते हो. जेनेटिक म्यूटेशंस आप देख सकते हो, जैसे कि यहाँ पे मैंने बताया. कैंसर के डिफरेंट फॉर्म्स आप कर सकते हो. फॉरेंसिक के बिना, लाइक कोई भी आपका सैंपल है, इफ यू वांट टू डू द डीएनए फिंगरप्रिंटिंग, राइट, सो वो आप यूज कर सकते हो. माइक्रोब डिटेक्शन एंड कैरेक्टराइजेशन के लिए, लाइक अननोन आपने बैक्टीरिया आइसोलेट किया, उसका आप 16s RNA एम्पलीफाई करके यू कैन नो कि विच बैक्टीरिया इज this. So these are all the applications of the PCR. So I think this is pretty much for the first part. If you liked it, hit the like button and do subscribe the channel for more such videos. And thank you so much for watching.